Welcome to all students. Myself, Om Prakash Godara, Assistant Professor, Principal of Seed Technology, Department, Faculty of Agriculture and Veterinary Science. JAYOTI Vidyapeeth, Women City, Jaipur. Today we discuss about our topic seed productions in cotton varieties and hybrids. Cotton, botanically as a Gossypium species, is a fiber yielding crop. So it is also known as clone of fiber crops. It serves as a cash crop to farmers as the lint serves the raw material for the textile industry. The seed is used both for multiplication and as animal feed. The success of commercial crop depend on the quality of the basic seed. Floral biology of cotton crop mainly simple, solitary and terminal extra axillary petals yellow to cream in color. Hermaphrodite and bracteoles called as epigallix and three in numbers. Three and deeply serrated and persisted at the base of the flowers. Nectary glands is present on each bracteoles and calyx five united. Cup-shaped corolla five and polypetals. A purple spots present on the inner side of the claw of the petals. Is like petal spots in some species. Androecium forming staminal columns, monodelphous, bearing numerous centers, ovary superior pentacarpellary and style slenders, passes through staminal columns with 3 to 5 lobed stigma. Ovals many in exile placentation. mainly स्टिग्मा टॉप पार्ट्स पे प्रेजेंट होती है उसको जस्ट नीचे मेनली जो स्टाइल्स है और स्टाइल्स जो मेनली है वो मेनली स्टिग्मा से फिलामेंट्स के द्वारा कनेक्ट होती है और जस्ट बिलो मेनली एंथस है वो प्रेजेंट रहता है वो जो ओवरी है वो मेनली इंटीरियर पार्ट पे और जो मेनली पेटल्स है वो टॉप पार्ट पे प्रेजेंट है. There is much variation in case of flower openings. Asiatic cotton opens between 8 a.m. to 10 a.m. मॉर्निंग के अंदर जो 8 a.m. से 10 a.m. तक में जो एसिटिक कॉटन है उसके अंदर जो फ्लोर ओपनिंग है वो होती है. अमेरिकन कॉटन ओपन मच अर्लियर. इसके अलावा जो अमेरिकन कॉटन है वो मेनली अर्लियर ओपन होती है. टेम्परेचर अफेक्ट द फ्लोर ओपनिंग मेनली टेम्परेचर flower opening को affect करता है. After flower opening, the cream yellow color of corolla turns pink within a day and later changes to red. Color changes इसमें होता है during daytime. The respectively of the stigma is 8 to 10. Cotton is an often cross-pollinated crops. मैंने जो cotton से often cross-pollinated crops होती है, where the extent of cross-pollination is greater than 60%. In cotton, four different species are in popular uses, mainly different four species. उसका maximum use किया जाता है. Is like Gossypium arboreums and Gossypium herbaceums, Gossypium hirsutums, and Gossypium barbadense. Method of seed production mainly जो seed production method है इसमें firstly जो varieties है under isolations by open pollinations the variety are multiplied. For nucleus seed production, shelving of flower is done with cotton and dipped in clay or red earth. Hybrids in cotton both inter and intra-specific hybrids are available. So, inter-specific hybrids is like Vara Lakshmi. It is close between Lakshmi into SV298 Gossypium hirsutums mainly and cross between Gossypium barbadense. TCS 32 और या जया लक्ष्मी इसका जो मेनली कोर्स है वो DS28 इसका क्रॉसिंग मेनली SP42 से करवाते हैं जिसका सर्टिफिकेट नेम गॉसिपियम इंस्टीट्यूट एंड गॉसिपियम बारबाडेक्स है. TCH B213 इसका TCH 1218 इसका जो क्रॉसिंग है वो मेनली TCB 209 से करवाते हैं. इंटर स्पेसिफिक हाइब्रिड कैन बी मेनली सुगुना सविता और इसका सविता है जिसका मेनली क्रॉसिंग है वो T7 की क्रॉसिंग से M12 के साथ करवाई जाती है। टूल एम्प्लॉयड फॉर हाइब्रिड हाइब्रिड के अंदर मेनली जो प्रोडक्शन है उसके अंदर कौन कौन सी टूल से उसका यूज किया जाता है। द हाइब्रिड सीड प्रोडक्शन इन कॉटन इज अचीव थ्रू emasculation एंड टेस्टिंग टेक्निक्स which is the physical removal of male organs। तो मेल organs है उसको मेनली फिजिकली रिमूव करते हैं। स्टेमिनल कॉलम्स को है मेनली रिमूव किया जाता है एंड फ्रॉम द फीमेल पेरेंट्स जो फीमेल पेरेंट्स है उससे मेनली जो मेल जो पार्ट है उसको रिमूव किया जाता है। emasculations and testings at the time of flowering initiations। जब flowering initiations start होते हैं in female lines, the flower that are going to often next day are selected and the petals are removed between 3 to 6 p.m. इवनिंग टाइम में मेनली जो पेटल्स है उसको रिमूव किया जाता है विद द हेल्प ऑफ नेल और नीडल्स, या तो नाखून से या मेनली जो नीडल्स है उसका यूज किया जाता है। द टोटल स्टेमिनल्स पोलन प्लस एंथर ट्यूब्स। स्टेमिनल्स के अंदर मेनली थ्री पार्ट्स इन्वॉल्व होते हैं, पोलन और एंथर एंड एंथर ट्यूब्स। पोलन आर रिमूव्स, ये जो मेनली मेल पार्ट है इसको रिमूव किया जाता है। देन द फ्लावर आर कवर्ड विद डेफिनेट कलर कवर और इजी आइडेंटिफिकेशन से इसको एक अपने पास टैगिंग होती है उसके द्वारा एक स्पेसिफिक टैगिंग के द्वारा इसको मेनली टैग किया जाता है कि इसका जो आइडेंटिफिकेशन है वो अपन इजी वे कर सकते हैं। Of the emasculated flower in the morning between say 9 a.m. to 12 noon, morning 9 a.m. से 12 noon, which is the anthesis time। So ये एक anthesis का times होता है। The flower of selected male parents are plucked and dusted on the stigma of the emasculated flowers on opening the course। mainly उस cover को open करके और जो open को portion करवानी है उसका जो mainly anthers है उसको mainly और जो stigma part है उसको mainly dusting किया जाता है। It is again covered with different color cover to avoid pollinations with other pollen and to identify the emasculated and dusted flower from the rest। The pollen from a single flower is enough to dust 4 to 5 to female flower। एक सिंगल जो फ्लावर्स होता है उससे जो मेल फ्लावर्स होता है उसकी जो डस्टिंग है वो 4 to 5 फीमेल्स के लिए अपन यूज कर सकते हैं। द पोलन रेस्पेक्टिवली ऑफ द स्टिग्मा इज फॉर 46 hours। For easy identifications of self-head wall from emasculated and dusted wall, the bract can be removed while emasculating on to the little contributions of bract of seed to shade the seed yield। पर्टिकुलर ऑफ वैरायटी एंड हाइब्रिड से मेनली जो डिफरेंट वैरायटीज होती है उसके अंदर जो क्रॉस है मल्टीपल क्रॉस किसके बीच में करवाया जाता है वो जो सीजन्स है कौन सी सीजन्स उसके लिए बेटर होती है और इरिगेटेड एंड रेनफेड कंडीशंस होती है या उसका सीड यील्ड मेजरमेंट्स कितना होता है। तो जो मेनली वैरायटी है MCU 5 है वो मेनली इसका जो पेरेंटेज है तो पेरेंटेज का मतलब मेनली जो कौन कौन से पेरेंट्स के बीच में मल्टीपल क्रॉस के द्वारा जो इसको प्राप्त किया जाता है। और इसका जो सीजन है वो मेनली अगस्त जनवरी होता है। इरिगेटेड कंडीशंस के अंदर ये जो कॉटन से वो होती है उसकी जो सीड यील्ड है वो 18,850 kg/hectare से। MCU 7 से इसका जो मेनली क्रॉस है वो मेनली एक्सरे इरिडिकेशन से इसका जो क्रॉस है L1143 एक जो वेराइटीज है उसके साथ क्रॉस किया जाता है। और ये वेराइटीज मेनली जनवरी फरवरी टू मई या जून समर से उसके अंदर मेनली इसका सीजन फ्रूट्स होता है और इरिगेटेड कंडीशंस के अंदर मेनली होता है। इसकी जो यील्ड होती है वो 1333 kg/hectare होता है। इसका जो डिफरेंट वेराइटीज है उनका जो डिफरेंट सीजन्स होता है या इसका जो क्रॉस है मल्टीपल क्रॉस किससे करवाया जाता है उसकी यील्ड कितनी होती है। हाइब्रिड्स के अंदर मेनली जो स्विंस है ये हाइब्रिड डेरिवेटिव्स फ्रॉम द क्रॉस सुजाता एंड विंसेंट्स के बीच में मेनली क्रॉस करवाया जाता है और उसका मेनली जो सीजन्स होती है अगस्त फरवरी होती है इरिगेटेड कंडीशंस के अंदर होता है और उसकी जो यील्ड होती है वो 1020 kg/hectare होती है। जयलक्ष्मी है ये एक इंटर स्पेसिफिक हाइब्रिड्स ऑफ DS28 गोसिपियम हिस्टोटम एंड SP4 to 5 मेनली गोसिपियम बारबाड उसके बीच मेनली क्रॉस करवाने को मेनली जयलक्ष्मी हाइब्रिड्स है उसकी अपन को फाउंडेशन हुई और इसका जो सीजन है वो मेनली अगस्त फरवरी होता है और ये इरिगेटेड कंडीशन के अंदर होती है और इसका जो यील्ड है वो 2880 kg/hectare होता है। So steps in hybridizing techniques. तो hybridizing techniques है उसके अंदर कौन कौन से जो steps है उसको use किया जाता है. तो emasculated and dusted is a far possible birds upbringing during the first 6 weeks of reproduction phase to ensure good setting and development of balls. Restrict your emasculations each day evening from 3 p.m. to 6 p.m. तो mainly जो evening times होता है उसके अंदर mainly जो emasculations है वो अपन को नहीं करना चाहिए, restrict रहता है. And pollination in morning between 9 to 12 noon to ensure highest purity of hybrid seeds. तो पोलिनेशन होता है मॉर्निंग टाइम में 9 to 12 noon. तो मेनली यहाँ जो अपनी हाइब्रिड सीड सेटिंग की कैपेसिटी है और या प्योरिटी है वो बहुत ही ज्यादा होती है. मस्कुलेशन शुड बी कंप्लीट एंड परफेक्ट होता है. Choose optimum size of bird and avoid young or too old birds for musculation. So mainly जो musculation है उसके लिए जो optimum size की जो birds है and avoid young or too old birds. इसके अलावा जो young और जो old birds है उसको mainly remove करना चाहिए. कवर द मेल बर्ड्स विद पेपर बैग्स. मेनली जो मेल बर्ड्स है उसको मेनली पेपर बैग्स के द्वारा कॉन्जेक्ट किया जाता है प्रीवियस इवनिंग फॉर देयर यूज नेक्स्ट डे इसको यूज के लिए. एंड emasculated birds may be covered preferably with butter papers. Do not forget to the tie threads to the pedicles of the birds immediately after pollination. After pollination के बाद में जो pedicles से जो threads है उसको मेनली अपन tie कर देते हैं. Close your crossings programs after ninth week from commencements of crossings and remove all birds and flowers appearing subsequently to facilitate the development of cross wall. Nip the top and side shoots to stop further vertical and horizontal growth. Light irritations should be given and when required, say, excessive or scanty or inadequate irrigation should be avoided, especially during crossing and ball development speeds. तो mainly ball development speeds के दौरान mainly जो irrigation है वो proper होनी चाहिए. Continue irrigation till last picking of the crossed balls. Frequency of irrigations depend on weather factors like rainfall, temperature, and wind velocity. Pick up the ripe and completely opened balls along the with threads and collect in baskets for second sorting. उसके बाद में mainly अपन को जो ripe completely opened balls है उसको pick up कर लेना चाहिए and after that इसको storing sorting कर लेना चाहिए. Balls without thread में भी bulk harvested as female sheet cottons. Crossed balls collected in baskets may be sorted out from second times to verify that they are crossed balls. Then collect crossed seeds, cottons and store in gunny bags, carefully marked as crossed balls. So, उसको mainly marking करना भी जरूरी है. Rain touch cottons or hard locks be picked and kept separately to avoid poor germinations of hybrid seeds. तो hybrid seeds इसके अंदर poor germinations हुआ उनको अपन को अलग से कर देना चाहिए. Store the cross seeds cotton in cool dry place till it is handled over to processing units. Use of genetic marriage sterility. Mainly hybrids are also produced by employing genetic marriage sterility systems in cottons, where the females parents will segregate into 50-50 ratio of male steriles and male fertile plants. Male fertile plants are removed and the male sterile plants are processed with the concerned smell lines. Land requirements, the field should be fertile and form into rich and furrow. Black cotton soils are highly preferable than other soils. Land should be free from volunteer plants and designated seed disease, especially the wind disease. Seasons mainly winter crops August to September and summer crop February to March. Seed and sowing seeds should be obtained from an authenticated source with the tag and bits. Pre-sowing management says the seed can be hardened with the 1% prosopis and pungum's leaf extract for rainfed or summer sowing to resist water stress problems. Use of deleted seed is better than fuzzy seeds to avoid disease and injured seeds. Seeded mainly varieties 15 kg per hectare fuzzy seeds and 7.5 kg per hectare deleted seeds and hybrids 3.75 kg per hectare Jayalakshmi and 1 kg TCBH 213. Male 2 kg per hectare and females 4 kg per hectare. Seed treatments treat the seeds with the azospirillum at 3 packets 600 grams per hectare and 2 kg of azospirillum per hectare mixed with 25 kg of FIM and 25 kg of soils and applied on the seed lines. These save 25% nitrogen beside increasing yield. Spacing mainly different variety के अंदर जो spacing है वो कितनी होनी चाहिए तो long duration variety उसके अंदर rotor distance 19 to 30 centimeters and short duration varieties है उसके अंदर mainly 16 to 30 centimeter होती है and in hybrids mainly जो male है उसके अंदर 120 to 60 centimeter होनी चाहिए और short durations जो female है उसके अंदर 90 to 60 centimeters जो spacing है वो requirements complete करनी चाहिए. Hybrid planning ratio 8 ratio 2, but here is block systems where floor of two parts of male is sufficient to dust 8 part of female parents. तो mainly जो two parts males हैं उसको mainly 8 parents parts of two females हैं उसको mainly reproduce कर सकता है. आइसोलेशन डिस्टेंस है वो फाउंडेशन शीट्स और सर्टिफाइड शीट्स के अंदर जो मेनली वेराइटीज है उसके अंदर जो फाउंडेशन शीट्स की जो डिस्टेंस है आइसोलेशन वो 50 होती है एंड सर्टिफाइड शीट्स के अंदर मेनली 30 सेंटीमीटर 30 meters and in hybrids foundation sheet also 50 meters isolation distance and in hybrid certified sheets 30 meters isolation distance we kept. Manure and fertilizers mainly compost 12.5 tons per hectare and total consumption 100 ratio 50 ratio 25 NPK kg per hectare use कर सकते हैं. Vessel dose 50-50-25 NPK kg per hectare use करते हैं and crop direction only जो nitrogen है उसका 25 kg per hectare use करते हैं 40 to 45 days after sowing. 70 to 75 day after sowing mainly जो nitrogen है उसकी top dressing 25 kg per hectare use कर सकते हैं. foliar spray diammonium phosphate 2% for female parents and spray on 60, 70, 80 and 90 day after sowing. Sowing के 60, 70 and 80 and 90 day after mainly जो DAP है उसका 2% mainly अपन spray कर सकते हैं. So यहाँ सो के 5 kg of DAP and 25 liters of water overnight and super dense liquid should be taken and mixed with the 475 liters of water for spraying 1 hectare. इसका भी यूज कर सकते हैं. Micronutrient applications है इसके अंदर mainly mix 12.5 kg of micronutrient mixes formulated by the Department of Agriculture with enough sand to make a total quantity of 50 kg for 1 hectare. तो mainly जो micronutrients है वो according जैसे गवर्नमेंट डिमांड्स के अंदर जो रिसर्च के अकॉर्डिंग बताया गया है. Naphthalene acetic acid applications spray of 40 ppm of naphthalene acetic acids 4 milligrams of naphthalene acetic acid dissolved in 1 liter of water at 40 to 45 day using high volume spray liquids in 1125 liter per hectare and repeat the same dose after 15 days of first spray. Topping mainly topping arrest terminal growth by nipping the terminals 10 to 12th node for controlling excessive vegetative growth. जो excessive vegetative growth होती है उसको control करने के लिए mainly जो topping process है वो की जाती है and rogging the crop should be referred for all types self-fed plants from vegetative phase to harvest phase depending on plant status, leaf size, leaf color, hairness, stem color, flower color, petal spots, pollen color, number of symphodia, ball size, ball shape, and pittiness etc. to maintain genetic purity. Field standard mainly maximum permitted foundation seeds and in certified seeds of types of plants है उसके अंदर वो varieties के अंदर 0.1 होना चाहिए and hybrids के अंदर 0.1% and in certified seeds जो varieties है उसके अंदर जो type plants है उसकी जो percentage है 0.2 and in hybrids के अंदर 0.5% in certified seeds के अंदर जो है normal रहती है. Irrigation management Once in 10 days, critical periods are more formations to more maturation stage. Specific problems, ball shedding will occur either due to extreme dry climate or lesser frequency of irrigations और physiological disorder. ये इस कारण से मान लीजिए बॉल शेडिंग है वो होती है by spraying 40 ppm of naphthalene acetic acid and psychocells at 20 ppm. Then this can be minimize the ball sheddings. Harvesting. The seed attains physiological maturations 45 days after anthesis. The initiations of hair line crack on the dried bolls are the physical symptoms of physiological maturations. At that times, the moisture content will be 30 to 35%. The boll are harvested as it became important. Due to continuous flowering habit, once our harvested is not practiced, as and when the bolls brushed with hair lines cracks, the bolls are collected and dried. Normally, 5 to 7 picking can be practiced in a cotton crop, but early 4 to 5 picking are recommended for seed purpose. Harvesting in the morning hours up to 10 to 11 a.m. only when there is moisture so that dry leaves and bracts do not stick to the kapas and lower the market value. Pick kapas from well brushed balls only. Remove only the kapas from the balls and leaves the bracts on the plants. Uh, as kapas is picked, sort out good puffy ones and keep separately. Keep stand discolored and insect attacks kapas separately. Kapas sorting. Mainly kapas sorting is manually to pick a good quality seed. Hard locks are to be removed. Kapas without proper brusting and line is light yellow in colors. Since these kapas mostly result in poor quality seeds due to wall worms and other insect attacks. Skewed walls or ill fillets or non-viable seeds are formed is stigmatics lobes are not polluted. Greening and certifications in the cross capacity in separate gains erected in authorized seed processing units or farm gains under the close supervision of the authorities concerned to the ensures purity and avoid seed damage. Sieve the seeds in two types of mess and remove small shriveled seeds, broken seeds, and clean perfectly from any direct or dust. After cleaning, the seed should be dried well and cleaned by hand picking. After cleaning, certifications agency will take a sample for testing germinations and genetic purity test. Minimum germination percentage 65% and genetic purity 90% should be maintained. Certified seeds would be bagged in 1 kg bags, sealed and details regarding its origin, germination, physical purity percentage, and genetically purity percentage. Besides, seasons of productions are passed on to sale agency or respective producer or commercial sales. Uncertified seed would be procured by the concerned departments of agency at the market rate for the ordinary cotton seeds for further multiplications. This step is essential to avoid unauthorized sale of substandard uncertified seeds. Processing the grenade seeds or the fuzzy seeds are graded by hand picking and by pressing on wire mesh seeds to remove the undersized seeds or dust. Acid delinting fuzzy seeds will clog with one another, so for easy handling, the seeds are delinted using S2SO4 at the rate 100 ml per kg seed for 2 to 3 minutes. After acid treatments, the seed should be washed thoroughly for 3 to 4 times with fresh water. From the floaters, mature seeds without any visible damage can be picked and added to the sinkers. Weight quantity of fuzzy seeds is taken in plastic containers and required quantity of the acid is added. Stir wells with the wooden rod till shiny black colors appears. Wash with a more of water 5 to 6 times and set dry the seed to reduce the moisture contents to 12% before further handling. Processing of delinting sheets, the free flowing delinted sheets can be graded using 10 upon 64 round perforated metal sieves, which is recommended as standard sieves in OSW cleaner cum graders from cottons. The seed can also be graded by specific gravity methods by using producing techniques using water. The seeds will separate into floaters and sinkers. The sinkers are good seeds from floaters. Reddish images and damaged seeds with insect holes are removed. The brownish seeds, which are good seeds and hand pickets and used for sowing. Seed standard mainly characteristics physical purity in foundation seed 98% minimums and certified seeds also 98% and inert material maximums 2% in foundation seed and also 2% in certified seeds. Other crop seed mix maximum 5 seeds per kg in foundation seeds and 10 seeds per kg in certified seeds. Wheat seeds maximum in 5 per kg in foundation seeds and 10 per kg in certified seeds. Genetic purity percentage mainly foundation seed 100% and also certified seed in 100%. Germinations percentage in variety 65% in foundation seeds and also 65% in certified seeds. Germinations for hybrids minimum mainly 75% in foundation seeds and 75% in certified seeds. Moisture contents percent mainly moisture previous 10 mainly in percentage foundation sheets and certified sheet also 10%. Moisture vapor proofs mainly 6% in foundation sheets and also 6% in certified sheets. Seed can be stored up to 8 to 9 months in moisture previous containers and up to 12 to 15 months in moisture vapor proof containers. The seed treatment with thyrums activated 2.5 kilograms or chlorine-based halogens, which are said rate 3 kg, will protect the seeds from storage fungi, aspergillus species, and preserve the storability. Mid-storage corrections, the fuzzy and delinted seeds can be soaked in double the volumes of 10 kg minus 4 molar solutions of sodium hypophosphate for 2 and 1 horse respectively. Then the seeds are set to and sun dried to bring back to moisture content to 12%. So the procedures of seed production is important. Different जो techniques है उसका अपन use कर सकते हैं वीडियो एनालिसिस है या तो क्लोज है अपना जो मेल पार्ट है उसके अंदर जो इमास्कुलेशन प्रोसेस है किस प्रकार करनी है उसके अंदर जो मॉइस्चर कंटेंट या फिर जुगल मेच्योरिटी है उसको किस प्रकार आइडेंटिफाई किया जा सकता है तो उसके अकॉर्डिंग जो टेक्निकल प्रोसीजर है उसका अपन यूज करके और जो कॉटन है उसका बेटर हाइब्रिड सीड प्रोडक्शन कर सकते हैं